mentioned, I'm Soumya. I'm a PhD candidate in Feng Zhang's lab at the Broad Institute. And today I'll be speaking to you about some of our very recent work kind of exploring natural sequence diversity for discovering new kinds of programmable systems that we can use for kind of biotechnology development. Excuse me. So first, I just kind of want to define what a programmable system is. So when we think of programmable systems, we're kind of trying to identify systems that kind of are able to target specific biomolecules in a specific way, so that and in a way that we actually understand that targeting mechanism, and we can actually engineer it in order to change what that target specificity might be. And this is something that's very useful for therapeutics because, you know, if we're trying to perform some intervention at a specific biomolecule of interest, these kinds of systems allow us to specifically target that biomolecule and perform some type of intervention in order to prevent some kind of disease pathology, for example. So to focus a little bit more today, I'm going to be talking mostly about reprogrammable RNA-guided systems, which have proved extremely useful for kind of interventions at the genetic and transcriptomic level. One really great example of this that Professor Sharp already kind of explained in a lot of detail, so I won't really go into it, is RNA interference. So we would consider this to be programmable because by just changing the sequence of this small interfering RNA, we can change the target gene that is kind of being targeted and degraded by this natural RNA interference mechanism. Another really prominent example of this is CRISPR systems. Again, Professor Anderson kind of gave a really great background on how these work, but just to reiterate a little bit, since I'll be discussing this in more detail, CRISPR systems are bacterial adaptive immune systems that use these short guide RNAs that we also call CRISPR RNAs to target either DNA or RNA substrates in a specific manner and cleave them. And so this type of technology has been co-opted for DNA and RNA editing, as well as changing gene expression or modulating transcription. So the most dominant of these, again, which Professor Anderson kind of touched on, is Cas9, which is a nuclease that again uses guide RNA or CRISPR RNA to target a specific double-stranded DNA of interest. And in addition to this RNA guiding property, one of the things that's actually really special and useful about Cas9 specifically compared to other kinds of CRISPR enzymes is that it uses two separate nuclease domains to cleave each different strand of the DNA. And because of this modularity of these Cas9 enzymes, we can actually inactivate each of these domains specifically to create Cas9 nicases, which can specifically cleave one strand or the other, or both, or neither as well. And so this opens up a lot of different kinds of ways that we can interact with DNA substrates, including creating base editors, and prime editors, which allow for single base chemical modifications to a DNA substrate. Sorry. Yes, please do so. Sorry. Stress is not really made for the microphone, I guess. So I guess just to reiterate from the interruption, using Cas9, we can inactivate either nuclease domain specifically, and that allows for different kinds of ways that we can interact with DNA, and these are useful for different kinds of genome editing modalities that are useful therapeutically. So again, as highlighted by the other talks, one of the main problems with genome editing is not really our ability to edit genomes, but actually being able to deliver them for therapeutic use. So when we think about genome editing specifically, there's really two kinds of modes of therapeutic genome editing. One is ex vivo, where we take someone's cells out of their body, edit them in the lab, and then pick the ones that got edited correctly and put them back into their body. So a great example of this is CAR T cells, where we take people's cells out of their body, introduce some kind of synthetic T cell receptor, and then pick the ones that got the correct T cell receptor and put it back so that it can perform the therapeutic function. However, kind of like the dream of genome editing is that we can actually edit someone's genome inside of their body. And so this is really useful for a lot of different kinds of genome editing therapies, because there's a lot of diseases that people have where you can't take their cells out of their body and edit them. For example, with neurological diseases, you can't just take someone's neurons out of their body, perform some editing function, and then put them back in. You need to be able to do that inside of their body. And so the main challenge with this is how can we get these genome editing reagents into a person's body and into their cells efficiently and into specific cell types efficiently. So people use a lot of different ways of doing this, including viral vectors, lipid nanoparticles, which we just heard a lot of detail about from Professor Anderson, and even DNA nanostructures like Professor Bhatta mentioned. I'm going to focus specifically on one particular problem that's very well defined in this space. So one of the most common reagents for genome editing and gene replacement therapy right now in FDA-approved drugs is actually still using adeno-associated virus, or AAV, which are small viruses that infect human cells but don't cause any diseases. And so also, as Professor Bhatta mentioned earlier, these viruses are very efficient at targeting specific cell types and getting internalized to deliver genes in vivo. The main limitation with them in a technical capacity is that they have a limit as to how much DNA you can put inside of a single capsid. And it's actually quite a small amount. So for efficient packaging, you can only put a maximum of about 4.7 kilobases of DNA. However, when we look at Cas9, for example, the typical Cas9 gene is about 4.1 kilobases. We also need to include the guide RNA to actually guide it to the site of interest. We need regulatory elements to express those genes once you get that virus into the cell. And then, if we wanted to add any of these extra things like base editors, prime editors, et cetera, as you can see from the very simple math, it becomes way too large to put into a single capsid. So again, a lot of really amazing work is being done to kind of engineer and look at different kinds of therapeutic delivery platforms. In our lab, we're really interested in kind of exploring natural diversity to understand what kinds of solutions might exist in nature. And so we actually kind of took a different approach. So instead of looking at kind of the delivery platform itself, we wondered, could we just miniaturize the cargo that we're trying to package into these capsids in order to make it more efficiently packaged and kind of easier to deliver. So we basically looked for proteins that look like Cas9 in all available publicly available sequenced bacterial genomes and metagenomes. And specifically, we're looking for enzymes that kind of have the same domain structure that I mentioned before, where you have these two different nucleus domains, which are represented by the two different blue colors, and they kind of act independently of each other. And what we found is that there's this very large class of proteins called ISCB, which has this exact same domain architecture and is about a third of the size of Cas9. Unlike Cas9, they're not found as part of prokaryotic defense systems, but rather they're found in mobile elements or transposons in bacteria, which are just selfish elements in bacteria that kind of maintain themselves and move themselves around. So people had previously hypothesized that these ISCB proteins were kind of the evolutionary ancestors of Cas9. And again, the protein sequence suggested that these proteins were also nucleases similar to Cas9, but actually no experimental study or evidence had ever been presented before to understand how they could function. And in particular, what we really wondered is, if this protein really is an ancestor of Cas9, does it also interact with RNAs in some way that might allow it to be actually also an RNA-guided system? So we performed a lot of analysis of hundreds of loci harboring these ISCB proteins, as well as a lot of RNA sequencing experiments to kind of uncover whether or not they're associated with a non-coding RNA. And what we found actually is that these proteins are, in general, associated with these very large structured and conserved pieces of RNA. And these are also non-coding RNAs, so there's kind of no evidence that they're translated into proteins, but rather they're likely interacting with the ISCB protein. And in particular, what we noticed is that there was always this kind of portion of the RNA which is like very highly structured and highly conserved, which is shown by the kind of gray shading here. But also on the kind of 5' end or the start of the RNA, there's always an unconserved region that's also not very structured, but is always present in this RNA transcript. And so we wondered, could that just be the guiding portion of this piece of RNA? So to test this hypothesis, we took one of these systems and just replaced that kind of blue sequence with a sequence that we knew, and we tried to target different targets that were complementary to this guiding region. And what we saw is that indeed that kind of blue portion actually is a guide, and so similar to a Cas9 guide RNA or CRISPR RNA, you can simply replace that sequence and target any DNA sequence of interest, provided that it is adjacent to a short sequence motif, which we call the target adjacent motif. And so here's just an example of one such motif for one particular ISCB. So again, if we have a target that contains the sequence targeted by the guide RNA, so that's indicated by these two numbers, as well as the target adjacent motif, then we can see that the DNA is getting cleaved. Without that target adjacent motif, there's no cleavage, and also without the target, you don't get any cleavage. And so we can do this for any arbitrary guide sequence that we want, as long as the target adjacent motif is present. And additionally, this activity is dependent on the presence of the RNA, so if we don't have the RNA, we don't get any cleavage anymore. And so this is effectively the exact same mechanism by which Cas9 works. So of course, we observed this property for many ISCBs, and we also characterized some additional biochemical properties that showed more in general that these enzymes are very functionally similar to Cas9. So we really wondered, whether we could actually use these for genome editing. So we surveyed a lot of ISCB variants for this purpose, and we found that there are actually some which can perform genome editing in human cells, which really suggests a strong potential for development of RNA-guided DNA targeting therapeutics using ISCB as a platform rather than Cas9. And so here I'm just showing kind of like the typical sort of insertion deletion profile that you might get from one of these ISCBs at just a random site that we targeted. And again, this is just using kind of the non-homologous end joining pathway that Professor Anderson also described, where we're just cleaving DNA and natural cellular repair processes happen that leads to kind of either insertions or deletions forming. So we get, sometimes there's a small insertion, sometimes some bases are deleted. We also tried this at tens of sites across the human genome and saw that while there's some varying efficiency, in general, we can target any sequence, again, provided that it's adjacent to the target adjacent motif. However, we do see that this efficiency is quite low compared to a lot of Cas9s, and so in our future work, we kind of hope to focus on improving the efficiency of these systems and then subsequently showing that they can actually be used as a kind of base platform for adding some of these decorators that I described before, like base editors, prime editors, and other kinds of technologies that are being kind of developed constantly that currently use Cas9 in order to create a more compact scaffold that can be delivered. So again, overall, we found that ISCB is a much more compact RNA-guided DNA nuclease than Cas9. And it also, again, contains these two distinct nuclease domains that act to cleave each different strand of the DNA, and therefore has similar functional properties to Cas9. It's also active in human cells, so with some further engineering, we hope that it has a strong potential for therapeutic development for applications where Cas9 might be very difficult to deliver. So I also mentioned before that these enzymes are found in mobile elements, and actually these are a specific class of mobile elements, not to get too jargony, but they're called IS-200-605, and these mobile elements actually also include different, or sometimes harbor different kinds of nucleases other than ISCB. And so we kind of wondered, are these other nucleases also RNA guided, and have we just kind of, discovered a class of systems that just are like a huge untapped resource of different kinds of RNA-guided nucleases. So we first examined, and I'll just cover two of those examples right now. So we first examined ISRB, which is a protein that is almost identical to ISCB, but lacks one of these two domains, in this case, the kind of brighter blue H&H domain here. And we wondered, since these ISRB proteins were also associated with this kind of similar, highly structured RNA, which had a putative guiding region on it. Again, if this ISRB protein could be guided by the same structured RNA to perform DNA cleavage. So we tested this hypothesis, and what we actually found was kind of surprising, but in retrospect really made a lot of sense. So what I'm showing in this gel right here is two images of the same gel, but imaged in two different channels. So each strand of this double-stranded DNA target is dyed with a different fluorescent dye, so we can see each strand as it gets cleaved separately. And what we found is that ISRB, unlike ISCB, is capable of performing RNA-guided DNA nicking, in which case only one of the two target strands is actually cleaved, but the other one remains intact. And so this kind of made sense in retrospect because it is simply missing one of the two nuclease domains responsible for cleaving one of the two strands of DNA. And so this kind of represents the first naturally occurring RNA-guided nickase enzyme that has been discovered, and may also have some use for different kinds of genome editing modalities. The second example is this protein called TNPB. So in fact, the majority of these IS200, 605 mobile elements harbor not ISRB or ISCB, but actually these TNPB proteins. They may or may not be evolutionarily related to the other two proteins that I described. We're still kind of trying to understand that. But TNPB is hypothesized to be the ancestor of Cas12. Also, some variants of that are known as CPF1, which many of you might have also heard of as another prominent CRISPR enzyme that's used for genome editing. So I guess just to summarize shortly, we also found that TNPB is guided by a very distinct class of these structured RNAs that differs in the structure to the ISCB and ISRB proteins, but is also capable of guiding the TNPB to perform double-stranded DNA cleavage as well as single-stranded DNA cleavage in a guided manner. So again, all three of these nucleases, ISCB, ISRB, and TNPB, are encoded in mobile elements, and they're all RNA-guided nuclease or nickase enzymes that use an RNA guide to cleave a DNA target. And there are actually millions of these proteins in sequenced DNA, and so this represents a huge untapped resource of genome editing tools, something I'm not showing, but more recently some work was published showing that TNPB can also be used for human genome editing in cultured cells, so both of these two classes of proteins together are a very large resource of possible RNA-guided genome editing tools. And so there's a lot of different kinds of directions that we can go in, but we're really interested in being able to develop these proteins as a platform for different modes of genome editing, including just gene knockout, gene insertion, and base editing and prime editing, as I described. But kind of in a more broad sense, this kind of highlights the power of mining natural sequence diversity to overcome engineering problems. And so just to build on this theme as a final point, I will quickly touch on some similar work studying Cas13 proteins. So Cas13, like Cas9, is a CRISPR enzyme, but rather than targeting DNA, it targets RNA substrates using a similar short CRISPR RNA. Cas13 has previously been engineered to serve as a platform for RNA base editing, by which I mean when we fuse this 8R2 deaminase domain protein to the Cas13, we can target a sequence of interest, and the 8R2 is capable of deaminating a specific base within that target sequence. In this specific case, it changes an adenosine base to a guanosine base that's read out, or sorry, an inosine base that's read out by the ribosome as guanosine, so we can effectively perform A to G single base editing at an RNA level. And we're really interested in RNA editing as a therapeutic modality because unlike genome editing, it performs edits on transcripts which are transient, and so that is kind of a way to perform non-heritable changes to genetic material. And that's potentially a lot safer than doing genome editing. And so again, these proteins are extremely large and they don't really fit into kind of traditional genome editing or transcriptome editing delivery vehicles like AAV. And so we again kind of performed a similar type of analysis where we just looked for proteins that look like Cas13 but are just smaller and might be more amenable to being delivered by AAV. And so what we found is not a new class of proteins, but just new types of Cas13 proteins, which are closely related to those that are known, that I've highlighted here in red. And they are essentially similar as far as we could tell initially, but were just a lot more compact and kind of just to the small enough size that they could be packaged into AAV vectors. And I'm going to focus specifically on Cas13bt quickly because Cas13b type proteins had previously been developed as a platform for RNA-based editing. So I think we're running out of time, so just to kind of go through quickly, what we found is that Cas13bt can support RNA-based editing. And when we packaged this system into an AAV vector, specifically the Cas13bt fused to the 8R deaminase domain, the kind of regulatory components required to express that, and then also the guide RNA, we found that we were able to achieve targeted editing using an AAV delivery platform in cultured cells. And so this was sort of a proof of concept that suggests that it's possible to deliver functional RNA-based editing cargo using a single AAV genome, and we kind of hope to continue developing that technology. So I guess just to summarize overall, these two kind of shorter stories that I told you both focus on kind of the discovery of compact proteins that might be able to replace proteins like Cas9 and Cas13 for therapeutic purposes. And that is for the reason that the compact size makes them easier to package in traditional delivery vectors like adeno-associated virus. I will also mention quickly that even though other kinds of therapeutic delivery modalities like LNPs, for example, are able to package larger cargos, in general, manufacturing and production of those kinds of reagents tends to be more efficient and pure, the smaller that cargo is. And so this kind of miniaturization of these reagents can be helpful for engineering different kinds of therapies, whether they're delivered by AAV or other modes. And so also this work kind of just highlights the power of looking at natural sequence diversity to solve problems that we might kind of traditionally approach through direct engineering of that system through protein engineering or directed evolution or some other kind of more complex and laborious process. And this is a theme that in the lab we're kind of really excited about continuing to explore and expand beyond just looking at miniaturization of systems to discovering totally new types of chemistries and other systems. So with that, I'd just like to thank the many people that contributed to this project. I'll specifically point out Han, who's the co-collaborator on all of the work that I presented today, Feng, who is my advisor and has been a wonderful and supportive mentor over the past years. We have some really great collaborators at Montana State University and the NIH who helped a lot with this, and also my thesis committee, including Professor Jim Collins, who will be presenting to you right after this, and our various funding sources, and also thank you to all of you for your time and attention. And happy to take questions.
Thank you, Soumya. That was terrific. When will Feng let you graduate?
I think soon, hopefully.
We have some time for some questions.
Hi, great talk. I was curious about these systems. Mm-hmm. Can you compare them with the current CRISPR-Cas9? Because they also have like families, right? So can you talk more about like the off-target effects, cutting efficiency, and immune responses if you got a chance to test those?
Yeah, so I'll touch on the first two. We haven't really looked at the immune response yet. This is still kind of a very early stage. We really only characterized the mechanism of these proteins like 8 months ago. But in terms of the kind of cutting efficiency and specificity, so what we found is that for ISCB, the efficiency of cleavage is much lower than Cas9 at the moment. If we compare to like the standard Streptococcus pyogenes Cas9 that most people use, it's also a lot less specific. And so one of the kind of projects that we're undertaking right now is to kind of do a more thorough job of surveying this natural diversity to try to find variants that might have kind of more relevant properties in that area. TNPB is more on par with the efficiency of Cas9 in terms of cleavage efficiency, and we have yet to survey the specificity of those. So yes, there's certainly still some work required, I think, to really bring them into like the parameter space that would be useful for a therapeutic application.
Okay, and then another question is, I've noticed that ISCB was around like 400 base pairs, which is really small and good, but for the long non-coding RNA, there's a lot of variance that ranges from like 1,000 to 10,000 base pairs. So I was curious, like, what's your average size of the long non-coding RNA that you're currently using for these systems?
You mean the guide RNA that goes with it? They're on average about 300 base pairs or so. So they're quite a lot larger than the kind of single guide RNA that people typically use with Cas9, probably about 3 times the size. But there's, again, some variation in that. So there are some variants that use kind of smaller and slightly longer versions. And also one of the things we're interested in is trying to minimize that RNA component by just both trying to examine the system structurally and then also performing kind of like mutagenesis experiments to try to minimize the size of that RNA.
Great. We have time for one more question. No questions? Oh, okay.
Thank you. Thank you very much for your talk today. I'm curious, the sort of the high level objective to this. You're clearly exploring a lot of space, but I'm wondering if is part of the objective to have a toolbox that others can use in a more prescriptive kind of way?
Yeah, I think in terms of engineering these specific systems that I discussed today, that really is, the goal is to kind of like build them into a toolbox that people can use in a prescriptive way, as you just said. I think more broadly, the goal of doing like these big kind of natural sequence diversity searches is more to uncover different kinds of chemistries that might be interesting, which is still ongoing. So unfortunately, I'm not really able to talk about it now. But I think, these kind of projects are sort of a proof that it's possible to kind of search for and find like specific types of proteins and characterize their mechanism in a detailed way. And we would hope to be able to expand that type of platform to look at not just RNA-guided DNA cutting enzymes, which we already have lots of, but different ways that we might actually interact with a DNA substrate, like transposition or DNA insertion, different kinds of things like that as well.
Thank you so much, Soumya.